All right, in this video we're going to talk a little bit about how do muscle cells, muscle fibers contract. So let's first talk about the signal. So in order for muscle fiber to contract, it needs a signal from the nervous system. So that signal is going to travel down that motor neuron and it's going to cause acetylcholine, the neurotransmitter, to be released into the synaptic cleft. And so as you can see in the diagram, there's vesicles at the end of the motor neuron that store acetylcholine. And when that signal gets down to the end of that, it's going to cause calcium to rush in. It's going to cause the vesicles to fuse with the membrane and release the acetylcholine into the synaptic cleft. Then that's going to diffuse across the synaptic cleft. It's going to bind to receptors on the motor end plate of the muscle fiber. That's going to generate an electrical signal inside the muscle fiber called a muscle impulse. Eventually, it's going to travel along the sarcolemma, down the sarcoplasmic reticulum, into the cisterna of the sarcoplasmic reticula, and then it's going to travel down into and cause different changes to occur. So there's something called excitation contraction coupling that occurs. When the muscle impulse gets to the sarcoplasmic reticulum, it's going to cause calcium ions to be released inside the sarcoplasm, the cytosol inside the muscle cell. That calcium is then going to bind to troponin on the thin filaments. It's going to cause troponin to change shape, which is going to then push tropomyosin out of the way, and that's going to expose the myosin binding sites on the actin. So now the cross bridges are going to form and going to bind the actin and myosin molecules together. The evidence that supports how we think this all works is called the sliding filament model. It has to do with what the changes that we see in the sarcomere when a muscle fiber is contracting. So when the sarcomere is shortened, the thick filaments slide past the thin filaments. And what we see is the H zone, which is in the center of the sarcomere, the area that's just thick filaments, gets narrower. The I bands also get narrower. And you can see that in the diagrams. The Z lines move closer together. So once the myosin cross bridge is attached to the myosin or to the actin binding site, now we've got the two fibers connected. Then the myosin cross bridges are going to go through what's called the power stroke, and they're going to pull the thin filaments in. This requires ATP. So ADP and phosphate then are released from the myosin, and then a new ATP is going to bind to the myosin. That's going to break the bridge between the actin and myosin and allow the muscle fiber then to relax. So ATP is used to contract and relax the cell. And this is why, or this is how the things go back to the normal position. You can see all this in the diagram. You can also see the diagram in your textbook. So in order for the muscle cell to relax, we've got to stop that signal. So to stop the signal, first of all, we've got to get rid of the acetylcholine that's in the synapse. And one of the ways we do that, there's an enzyme called acetylcholine esterase that breaks down any acetylcholine to stop the signal from the neuron creating the muscle impulse. Once that happens, there's no longer that signal. The calcium then moves back into the sarcoplasmic reticulum. Actin and myosin, once that cross bridge is broken, they can no longer rebind because there's no more calcium to move that out of the way. And this explains how rigor mortis occurs. So in rigor mortis, the muscles are going to contract because there's some remaining ATP in the cell, and calcium is going to leak out of the cell. But once the ATP that's left in the cell is used up, the cross bridges can't break, and so the muscles contract and they stay contracted for a period of time, and then eventually those cross bridges will just break down on their own. So this table 9.1 in your textbook summarizes all the steps in muscle contraction relaxation. I'm not going to go over all that again, but it's a good thing to look at as you study for your test.